All right, so our first speaker is joining us virtually. Hope I'm pronouncing this correctly. Dr. Nicole Coufal is a physician scientist and associate professor at the University of California, San Diego. She is also PI at Coufal Lab, where she and her team study how microglia contribute to neurodevelopment and neurodegenerative diseases, as well as exploring how to harness the power of microglia for potential therapies. Today she's going to be sharing insights from her research, comparing and contrasting microglia versus microglia-like cells, and what those differences might mean when translated into potential therapies. Dr. Coufal, welcome.
Thanks so much for having me, and I'm sorry I can't be there in person. Can you hear me? I think is the first question. I can't see anybody except an empty stage, so you'll have to tell me if the sound is working. Maybe?
Yes.
Yes, okay, great. That's great. And then do you want me to share my slides, or do you have my slides up? Any chance?
Sure. Dr. Coufal, can you share your slides, please?
Yes, I sure can. Let's see. Oh, let me see. Did that work correctly?
Yep, we see you.
And you see the correct screen, hopefully?
Yes.
Yes. Okay, that's a win. Yeah, I'm so sorry that I can't be there in person. I'm actually in Switzerland, where it's like 9 o'clock at night and 100 degrees doing a series of talks. So otherwise I totally would have come in person. And I really, I know I can't see any of you, but please, I tried to keep this relatively short, and I would love to answer questions in general. And so just very briefly about me, I'm a physician scientist. I'm actually, my PhD is in neuroscience. My clinical work is in pediatric critical care. So I'm an ICU doctor for children. I got interested in neuroimmunology, or the immune cells of the brain, when I saw kids mostly with genetic diseases that hadn't been previously diagnosed. Mitochondrial diseases, other diseases get much worse in the setting of minor illness, and got interested in the intersection between the immune system of the brain and neurodegeneration. I don't have a lot of disclosures. I do some consulting, which is unrelated to what I'm going to talk to you about today. And so I also think a great random person to ask questions of, as such things go. I don't come with specific therapies for you all on this, but more kind of very much a general interest in helping people who have genetic diseases that involve microglia or innate immunity and lead to neurodegeneration. And I talked to Erin Sullivan a little bit before I gave this, kind of prepared this talk, and I'm really trying to follow what some of the interests were there in terms of trying to explain what some of the science is behind kind of immunology, neuroimmunology, and why, why it might matter and how it might impact some of the therapeutic choices without a specific kind of therapeutic in mind. Because again, I don't have any major disclosures here. So, I mean, as most of you I'm sure know, the brain has tremendous amount of cellular diversity. It's the organ in your body that has the most different kinds of cells. So there's this huge number of different cells in the brain. And I think, when I did my PhD, the immune cells were not something that was discussed anywhere along the way. And they're kind of embedded in there, but they're hugely important. So what are these immune cells of the brain? So they're the tissue resident macrophage. And when you think about macrophages, they're the innate immune cell that is in every tissue of your body. Let's see if I can get a laser pointer. Nope. So you have macrophages that go everywhere all the time. In the brain, they're called your microglia. In the liver, they're your Kupffer cells. They're in your lymph nodes, they're in your intestines, they're everywhere. And they're the first line of defense against viruses and bacteria, but then they also have a huge number of tissue resident roles. So depending on where they go, they do things that are very specific to that tissue. And that is true of microglia as much as it is of any other tissue resident macrophage. This is not just that kind of immune line of defense, but tasks that are very much specific to the tissue. And I think one of the things that makes microglia really unique is that they come from the early yolk sac. And so I'm gonna hope you guys can see my pointer. And that's very early in development, like 4 weeks of gestational age, 4 to 5 weeks. What you see is early myeloid cells, early hematopoietic cells. So from cells that are going to become part of your kind of blood system. So different tissue than the other brain cells that you have, like neurons and astrocytes. They go to the brain and they always self-renew. And so if you have some microglial depletion and there's any microglia left, they repopulate from their own niche. They don't come from the peripheral bone marrow. And that's kind of different from many of your other ones. Most of your others end up coming from definitive hematopoiesis. And so throughout your life, your other macrophages are repleted from your bone marrow, but generally it's been believed that microglia are very much different and unique, and that is important and contributes to kind of homeostasis of your brain, that they have tasks that other macrophages can't take on. And so kind of the teaching is that really it's from this very, very early development where you're getting yolk sac hematopoietic cells that come to the brain, like I said, very early in gestation, that end up becoming all of the microglia that are in your brain, and that really nothing much is coming from your circulation, from your blood, unless there's some inflammation. And so the kinds of things people think about where peripheral macrophages or monocytes are coming into the brain is strokes or multiple sclerosis, like something where there's inflammation and your blood-brain barrier has some amount of breakdown. And so I think one of the really important things is that humans and mice are really different. And when I did my PhD in neuroscience, I guess, I don't think anybody ever mentioned the immune system of the brain or talked about it, but also mice were the most common model for how we kind of thought about diseases of the brain. And there's a lot of differences between humans and mice in the immune system specifically. And so a lot of researchers who study leukemia or other peripheral kind of blood problems, immune cell problems, use humanized mice because of those differences. And that's not something that people in neuroscience have done quite the same way. And so I think the realization that there are really big differences between mice and humans has a lot of implications for trying to translate therapies from mice to patients, right? None of you are mice, and there's a lot of therapies in mice that haven't made it successfully into patients, and all of us want to see there be better therapies for patients. And so what are some of the differences between mice and humans? So if you think about it, mice are going to survive almost anything, Chernobyl, like any sort of catastrophe. And part of that is that their immune systems do work differently. And so they're much more tolerant of infection and low-grade inflammation than humans are. And so in terms of like toxins from bacteria, it takes 1,000-fold higher amount of toxin to get a sickness response in a mouse than a human. Mice have much more tolerance, so they can have some amount of like bacterial inflammation or bacterial kind of content in their blood and be okay with that and let that happen, versus humans are very much resistant, and that our immune system immediately jumps on any kind of infectious experience or anything that's out of the ordinary and tries to contain it. And that kind of really changes how you deal with inflammation. And part of how that kind of translates through the lifespan is that how your past infectious experiences shape your future immune responses is very different between the two species. And that makes a lot of difference over the lifespan of a human, which is much longer than a mouse, in terms of how that then figures into your risk for something like neurodegeneration. And we know that in people who don't have a genetic cause, that having a serious illness increases your risk for neurodegeneration, having something like an ICU stay. And so even for those folks who don't have genetic predisposition, there's some kind of connection between immune challenge and kind of that risk for neurological disease. This is kind of the data when I first got really interested in microglia and need to understand the actual data, but the idea is that microglia are really unique among these other macrophages. One of this is the RNA, and this is the epigenetics, but I hope you can appreciate is that the pattern that you see across many different genes or genomic locations throughout either active histone marks or transcriptomes. This is the microglia here. It's just the pattern's very different. Like they don't look like the macrophages from your liver or your spleen or your skin. And so they're quite unique. This is what they look like in the brain. The red is blood vessels and the green are microglia. And so they are supposed to tile across the brain and have all these kind of little ramifications and touch, like they each have their own kind of area that they monitor. And so it's not just that they are, attack infectious invaders, but that they really are a key part of modulating the synapses around them, the growth factors. They have a lot of jobs that they do. And so this is from like a really great review that was also not written by me, but some of the jobs that microglia do in the brain that are very much specific to microglia and not to other tissue resident macrophages. And so microglia help decide which synapses are kept and which synapses are pruned. The synapses that are more active are supposed to stay and get stronger, and the ones that you aren't using are supposed to get pruned over time. And the microglia play a key role in that. They help get rid of neurons that are sick. They help decide for neuroprogenitors what they differentiate into and where they go. If there's myelin injury, they help clean up myelin injury and process myelin. So there's a lot of important roles in the brain they have outside of the immune response itself. And what happens when they get inflamed, or how did they react? There's a number of they're actually quite flexible as cells go. What I've shown you in the last slide was what healthy microglia are supposed to do. But what happens when things are not going quite right and there's some kind of inflammatory stimulus? And so there's all kinds of different ways that microglia can respond. Some of which are helpful, some of which are hurtful, and some of which are helpful in certain contexts and really critical, and then hurtful in others. So it's a really nuanced field, and I think it's something where something as simple as like, well, if we just got rid of them, that doesn't work when it is really important to have an inflammatory response. But too much is also bad, and so it's a little bit of a Goldilocks problem, I think, for a lot of neuroimmunology. And so that there are things that happen to microglia with in the white matter, with aging, inflammation, neurodegeneration, and then also in a variety of these leukodystrophies, with ALSP, of course, being one of them, where the ability and to phagocytose and is altered, the damage-associated phenotype and inflammatory phenotype is altered. And so they're a key target in many neurodegenerative diseases, not just ALSP, because of those roles in immune surveillance, synaptic pruning, phagocytosis, lots of different areas. All right, so what are some of the therapeutic approaches as I understand them? And again, I'm a pediatric critical care doctor, take care of a bunch of kids with mitochondrial diseases and leukodystrophies. Those are the kinds of neurocritical care things that I do in my role as a neurocritical care director. ALSP, of course, is an adult disease, but this is some of the kind of things that are out there on the horizon, I think. And so, I think for many different monogenetic diseases, doing some sort of adenovirus type approach is in the works. And I think this has been a huge problem in the innate immune field, that really AAV is not very efficient at infecting microglia. All of these neurons are green, none of the microglia are green. So I think this has been a huge, a lot of AAVs are struggling in terms of efficacy. Some have some efficacy, some don't. some lose efficacy over time, but I think especially for things that involve the immune system, the virus has not been very good at getting into microglia. Microglia are really, one of their main jobs is to get rid of viruses. Like that's a core task for them. They're generally pretty good at it. So I think that role helps them get rid of therapeutic viruses as well, and kind of has made it difficult for microgliopathies. And I think there's a lot of questions about myeloid replacement. Is that through, trying to figure out what diseases you need to do myeloid replacement, how to generate some kind of safe microglial surrogate, how to deplete the host microglia, I think, in many diseases is an issue, and then how to introduce timely microglial surrogates. And so I think this is, a core challenge for ALSP, and the pathway to one of the main pathways to trying to get a therapy that's not a druggable therapy. I'm sure there are also some druggable therapies in the works, but again, this is not necessarily my field. So in terms of the CSF1R variants in ALSP, there's been a couple papers on this recently, but there's, I think, some reasonable data that microglial replacement has a good role, and that there's a lot of deficits associated over time with ALSP that can be partially ameliorated, at least in some patients, through microglial replacement. And so I wanted to talk a little bit about what some of the pathways of that are, and then just a couple slides. I kept it pretty short. I'm so happy to share more if people have questions, but I didn't want to make it too scientifically overwhelming. In terms of how, my lab has thought about how well peripheral monocytes can replace macrophages. And so, I think there's a lot of people who are interested in stem cell transplant as a potential therapy for ALSP. And of the two main ways that bone marrow transplant works is either as an autologous cell therapy, which is that taking bone marrow cells from a patient, correcting the genetic deficit in some way, and then giving them back to the same patient. And then the second way would be allogeneic cell therapy, and that is having a matching donor, and then taking their healthy cells and giving them to the patients. So they wouldn't have the genetic mutation, but they are then from a different donor that has to be matched on multiple points in order to make sure that the genetics are matched. Stem cell transplant, hematopoietic stem cell transplant, is the therapeutic modality of choice for a number of pediatric diseases. It's the standard of care. That includes things like X-linked leukodystrophy, with Scott Aldrich. So there's a number of different genetic diseases where if you have any neurological sequelae at all, the standard of care right now is to get some kind of a bone marrow transplant. And there's other ones where, it's currently in study and there are clinical trials for bone marrow transplant in these patients. That being said, this has a lot of potential issues and a lot of side effects, and is really not something that is taken lightly, taking care of a lot of these kids. And it's never good when you have to see an ICU doctor. So some of those issues, like if the patient is getting stem cells from somebody else, you can have graft versus host disease, where their immune system that has now populated the patient's bone marrow starts to attack tissues of the body. There's a lot of side effects that can come from the conditioning. So you have to, either way, if the stem cells are from yourself or they're from somebody else, you have to get rid of your own bone marrow to then put those in. And that is a pretty toxic process that can have a number of side effects depending on what medications are used. And I do think there's some new horizons of things that are coming out that will hopefully lead to less long-term side effects from conditioning, but a lot of the standard of care now still has a lot of side effects. I think the data for all of these diseases, and also for those people who have had bone marrow transplant from ALSP, is very mixed. So there's variable efficacy, and I don't think we quite always know why. I think there's still, I think this is put a plug for natural history studies. I'm sure somebody's doing it for ALSP, but it's so important to have have a natural history cohort and be able to say what the natural history is, and also to follow up from some of these therapies to try and figure out why some patients do well, and some patients don't, and some patients get worse after bone marrow transplant. And I think that's, a hugely scary thing, at least in the patients I've seen with things like X-linked leukodystrophy, and really, it causes a huge amount of consternation, rightly so, to families. And then, I think, we all like to think giving our own bone marrow back with gene correction is the best thing, but some of those therapies have had a huge number of problems. The most recent one is probably Skysona, which is the one for X-linked leukodystrophy, which has a 15% risk of malignancy with the viral correction before giving the bone marrow back. So, and now has a black box warning. So I think there's still a lot to be done in this field, and I'm glad we have a therapy and a potential therapy to offer families, but it still has quite a lot of issues associated with it. Like I said, those side effects are serious and they're long-term, and whether they're ones where the bone marrow comes from somebody else who doesn't have the gene mutation, or whether it's the patient's own that then has a lentiviral or AAV associated upregulation of the therapeutic gene. Both of those have had some pretty significant issues, and other clinical trials that have worked well. So I think it's something that needs to be, done in an investigative way to make sure we try to get the safest things to families. Okay, so I think I think one of the unanswered questions on that is how well, okay, if you decide to do a bone marrow transplant, if that's the standard of care, how well do myeloid cells that come from the bone marrow go to the brain and replace your microglia? It's kind of surprising to me, given how many of those diseases have bone marrow transplant as the standard of care treatment or investigative treatment, that we don't really know the answer to this very well. I think there's been one reasonable study on patients who've had bone marrow transplant where it was like a different sex between the donor and the recipient, and they could track the cells in the brain, and they looked in the brain afterwards, and it was something like 8 to 15% of the myeloid cells in the brain were replaced by the donor. So not a huge amount. So I think that's one question. But the second question is, like, how well do myeloid cells, when they do replace microglia, how well do they do that job? They're not meant to do that job, right? Like, it's really supposed to be microglia throughout your lifespan. And primarily until the last couple of years, this has been done in mice. But I already told you that mice are often not a great model for the humans. So I think assuming that it works the same in a person as it does in a mouse is really not a good assumption, and one that I would be very wary of. So this is probably the best paper out there on looking at mice and how well do mouse peripheral monocytes become microglia. They did a really beautiful job doing transplants. This is into the CSF1 receptor knockout mouse. And like, this is the microglia. This is if they used a fetal brain, like early microglia, and this is bone marrow or blood transplant. And this is looking at the genes that are different between them, the blue ones being things that come from the yolk sac that should make microglia, and the orange ones being things that are peripheral myeloid states. And I think overall, big picture, this list of genes is short. You have a lot of genes, and this is not a very big list, and the differences are pretty modest. So I think the general kind of assumption from a lot of the literature has been that, like, pretty good. Like, like you're not quite making microglia, but good enough for government work. This must be why it works. If we can just do better myeloid replacement, that will be fine. Um, and this is from a review, but really you can see there's only a couple markers that are different between them, and that was a little bit the theory of how this works. And I think, so my lab wanted to see if this worked in human. I approach a lot of things with skepticism. I think Dr. Blinton-Jones is giving a talk a little bit later here, so his lab did something, pretty similar in parallel. And really, I think the essential question was one of, if you put peripheral myeloid cells, either CD34 hematopoietic progenitors, which is what you would be using in a bone marrow transplant, or monocytes, which are what you would be getting from the blood, into the brain environment, how well do those become microglia? And what a lot of data in the field has shown is that there is a mouse model where you can do this in. It requires a lot of different genetics, so it's a little bit of a painful, complicated process to get the right genetic model. Basically, this model is humanized for MCSF, which is an essential growth factor. It's depleted of mouse microglia through a deletion in the mouse CSF1 receptor that takes their microglia away. And then it's depleted of adaptive immunity, which is what you have to do if you want to do cross-species transplantation of any kind. And so what we tried to do was to introduce either primary human microglia or those derived from iPSCs or CD34 hematopoietic progenitors or monocytes. And we've used CD34 cells both from adults and also from umbilical cord blood, which are supposed to be like the earliest possible hematopoietic progenitor that's used in bone marrow transplant. That's supposed to be the most permissive for HLA mismatches and like really like sought after for patients who are struggling to find a match, because it should be more primitive than other kinds of CD34 cells. And so we wanted to transplant all of these in the brain or peripherally and ask, like, how well do human myeloid cells become, peripheral ones become microglia in the brain, really trying not to rely on this assumption that it's going to work just like it works in the mouse. And what we saw here, and I kept it light, but I'm so happy if anybody wants to reach out to me later to send you a preprint or chat with you. Like I said, I don't have any major conflicts of interest in this area. I just really, like to love sharing our science and trying to get better things to patients in general. And so what you can see here is we use primary human microglia, those derived from iPSCs, CD34 cells, which is what you would use for bone marrow transplant, and monocytes. And this is a human-specific nuclei marker. Put them in the brain. We did RNA-seq, and what I hope you can see is that iPSC-derived microglia, which has been shown through a couple different papers already, but and was as we expected, are pretty close to primary microglia. They seem to follow that kind of yolk sac lineage. And what we saw from both the CD34 cells and from the peripheral monocytes is that they cluster very separately and they looked really inflammatory. And so some of the things that were big picture that are different was antigen presentation, which is part of recruiting B and T cells and causing inflammation, was much, much higher in peripheral myeloid cells. And then interferon response, which interferon is really it's implicated in aging, in like loss of cognitive abilities with aging in the brain, and having a huge interferon response in the brain, I think, is also generally considered deleterious. And we asked, like, how well does this compare to the mouse? And then also, what does this do to the brain over time? Like, is this a negative detractor to the brain? And what we saw, this is comparing the human and the mouse. And I hope what you can see here is that like none of this happened with peripheral myeloid cells in the mouse that make it to the brain. You don't get this huge interferon response. You don't get antigen presentation. Your your myeloid cells from the periphery, when they make it to the brain, are not angry, which maybe I think is related to that kind of tolerance versus resistance and how primed our own immune systems are to try and find invaders and get rid of them. And kind of briefly, just an over summary. Like I said, I kept this pretty short on the data because there's quite a bit more to not make it too overwhelming. But what we saw was that this was deleterious to the brain. So we did the transplants here really early, like postnatal day 2 to 5 of life. So this is pretty early in kind of the mouse's life. And then we looked when they were adults, just about 2 months, which is like when they would, start having their own litters, be or full adults. So a good amount of time. And what we saw was that the rest of the brain was not happy. We saw myelin loss, which I didn't put in here, astrogliosis, so the astrocytes are reactive. And then we saw loss of synapses across like several different ways. And that was both excitatory and inhibitory synapses. And then ultimately we saw the cortical thinning where some of the large neurons in layer 5 of the cortex were reduced. So like all of this seemed pretty detrimental to the brain. We found some correlates to this in some of kind of human studies with aging. So I think there's a lot there to kind of think about, and I think the data seems to suggest pretty strongly that peripheral myeloid cells in humans, it may be better than the myeloid cells that you have that have the gene mutation, that not having the gene mutation is beneficial, but that in the long term that they struggle to become microglia and that may be detrimental over time. So this is where I've landed in terms of unmet needs in brain replacement therapies. I think like trying to get a safe and effective brain myeloid replacement, be that from the patient themselves, from stem cells, whatever it is, I think is important to understand and to try to figure out. I think everybody would prefer it to be autologous from themselves. I think that that is definitely a goal for the field, and we all want minimal side effects. Maximal therapeutic benefit, minimal side effects. I think is the goal of every physician in taking care of their patients, trying to come up with something that helps that. I think some of the current hurdles is that potentially harmful nature of non-microglial myeloid cells to the brain long term. I think there's definitely, I have concerns after the studies that some of us have done that they can't become microglia, and that there's some really essential things we don't understand about the differences between peripheral and yolk sac myeloid cells that are really important to their function in the brain. I think a lot of the microglial depletion strategies in humans have not been clearly tested or well understood, like how to get rid of dysfunctional microglia in order to replace them. Looking at the safety and efficacy in more preclinical models, and iPSC microglia I think are a potential alternative, but this is costly and very time consuming. So I think there's a lot of space in the field for doing better, and I have a ton of collaborators to thank because my lab is a pretty good size. I don't do any of the work myself. Jing Wang here is the primary postdoc who worked on this project. Is really fantastic, and has done a really an amazing job. So I'm going to stop sharing, but I would love to answer any questions. I'm so sorry I can't see any of you all.
Now you can see everybody. Oh, no. Do we have any questions? I'm going to break this. Somebody know what they're doing?
This is Dr. Zolek, Mayo Clinic. Do you hear me, Dr. Coufal?
Yes, yeah, I do.
So in human studies, oh, by the way, very nice lecture. I enjoyed that very much. So in human studies, especially in radiological studies, usually cerebellum and spinal cord are safe. So listening to your talk, I was wondering what are the differences in microglia structure, function, morphology between microglia in the cerebrum and microglia in cerebellum and microglia in spinal cord? So in your mice studies, have you looked into these matters?
Yeah, that's a great question. We have looked into it a little bit because we're also really curious in how well transplant takes on niche specific phenotypes. And we've looked some at the cerebellum and the brainstem and the spinal cord microglia. I think there's still a lot of debate about whether, like, there's some studies in mouse that have suggested that cerebellar microglia may become a little bit later during fetal development and have a little bit different ontogeny. I think that has not been, like, it's still, I would say, controversial. I don't know, in my mind, it's still controversial. So I don't think that we quite know that. There's definitely some niche specific differences in the RNA and in the transcription. I think a lot of studies have struggled a little bit because most people do single nuclei studies from human spinal cord. And the problem there is microglia are only 5% of the cells in any given brain area. And so you end up with not very many microglia in a lot of these studies because they're so focused on other neuronal and myelin cell types. And so it's hard sometimes to get enough cells out of those data sets to be able to say something definitive about how they're not like structured to look at microglia, basically, I think, in that setting. So I do think we have some, like, the microglia take on some niche specific differences, but how well niche specific differences from the mouse translate to the human and what exactly those are and what they mean have not been necessarily well characterized or fully understood.
Do we have any other questions?
Sorry, I can only see myself.
Well, thank you very much for that presentation.